Dear students, today's continuation topic is mitosis in that prophase. Prophase main what are the characters are there? Here chromosomal material which untangled and condensed to form mitotic chromosomes. Mitotic chromosomes. And they are seen to be composed of the two chromatids attached together at the centromere. This is a centromere, two chromatids which are attached in the centromere part. Chromosomes begin to move towards the opposite poles. See, this is the opposite pole, here is the opposite pole. And each centrosome radiates out microtubule called as a ester. The two esters together with the spindle fibers form a mitotic apparatus. Here is the centrioles. This is the centriole and opposite pole it's there. It forms the thread-like structures which we called as the microtubules, which is nothing but spindle fibers. These together called as the mitotic apparatus. Right? This is the main character of late prophase stage. And here cell at the end of prophase do not show Golgi complex. This is very important. Should not show the Golgi complex and endoplasmic reticulum, nucleolus and nuclear envelope. See here nuclear envelope gradually disappearing. This is the main character of this prophase. And now the cell enter into the next phase is the metaphase. In the metaphase, what are the main characters we find here? Nuclear envelope completely disintegrates. See here, nuclear envelope completely disintegrates, and the chromosomes are spread through the cytoplasm of the cell. They can freely here is the no nuclear envelope. That's the reason they freely spread whole over the cytoplasm. And chromosomes condensation is completed. It was started in the prophase. That condensation will be completed in the metaphase. They can be studied easily under microscope. When we cut the onion root tip early morning, if you focus in the microscope, we can see the metaphase chromosomes clearly. They will have a two sister chromatids. Here is the chromosomes coming to lie at the equator. This is the equator. This plane is what is the blue color line what I drawn here. This middle of this one is the equator plate. In the metaphase, the chromosomes will be comes freely in the cytoplasm and they settled in the metaphase plate, nothing but equator. The plane of the alignment of chromosomes at the metaphase is called as the metaphase plate. Where they are lie the chromosomes we called as the metaphase plate. The spindle fibers there which are formed from the centriole. The chromatids their kinetochore of the chromosome they will be attached. See here, this is the chromosome, this is the centromere. To the centromere here is the small projection is there, which is the depression is there. This is called as nothing but kinetochore. Okay, kinetochore in the centromere. In this only, if you see that anaphase, now cell going to move into the anaphase. In the anaphase, majorly you can notice this is the shortest phase in the mitosis. Centromere of each chromosome divides longitudinally. Centromere of the chromosome, which is the chromosome previously it's like this, which are have two sister chromatids. Then they will longitudinally they should be divides. It form a two daughter chromatids, which is the chromosomes of the future daughter nuclei. 1 into 2. It will be as a longitudinal divides. Here is the one chromosome, here is the one chromosome. Got it? At the spindle fibers contract. Where it is the spindle fibers are contract in the ester. The chromatids move from the equator to the opposite poles. These chromatids they pulled to the equator opposite from equator to from here to here equator to opposite pole. In such way they will be pulled towards the both ends. Got it? And moreover the in this after anaphase the phase is nothing but telophase. In the telophase what are the main changes takes place? Chromosomes cluster at opposite poles. See, these chromosomes are this opposite pole or the opposite direction. They will be formed as a cluster and uncoil. In the prophase they coiled. Now in telophase they are uncoiled into chromatin fibers, which is there in the initial stage, very early stage. It has a chromatin fibers. Now nuclear envelope assembles around the chromatin, which is a prophase, early and late prophases. Very earlier, it is gradually disappear. The same chroma as the nuclear membrane will be appears here. Due to that reason, when it is around the chromosomes, this is formed, nuclear envelope. That's the reason thus daughter nuclei are formed. Two nucleus are there in the cell. Now here nucleolus and Golgi complex and endoplasmic reticulum will be reappear again. The spindle fibers there they will be disappear in this phase, in the telophase, right? And next one, see, I told that what is the subphases in the metaphase. First one is the prophase, metaphase, anaphase, telophase, followed by the cytokinesis. I given the code is the PM at Chennai, Prime Minister at Chennai. This is the order. Here is the last one is the cytokinesis. Cytokinesis is nothing but division of cytoplasm. See here, this is the division of cytoplasm. What will be happens that resulting in the formation of two daughter cells. Previously do daughter nucleus, two daughter nucleuses, which is the karyokinesis. Now cytokinesis means division of cytoplasm. When it is starts, when it's a telophase in progress, it starts when telophase in progress. Due to the cytokinesis, cytoplasm going to divide into two halves. Then two daughter cells are formed in this phase only. So here in animal cell I have a small difference is there in the plant cell. Because of plant cell have a cell wall, cellulose content cell wall. Animal cell have a only plasma membrane. Now here see here in the animal cell, cleavage furrow formed. This is called as nothing but cleavage furrow. Both ends like a some protrude, it will be enter. It's a gradually deepens and joins to center, divided the cytoplasm into two. Okay, it's a small invasion takes place. Due to that is gradually grows and the cytoplasm going to change into two cells. Got it? Whereas in the plants, in the animals, what will form in the animal cells? It's a cleavage furrow formed. Whereas in the plant cell, it's a quite different because of have a that cell wall is present here. It is different from the cytokinesis in animal cell due to the presence of cell wall, which is made of the cellulose. In plant cell, vesicles formed from the Golgi bodies. In the telophase itself only Golgi buds appear, the body cell accumulate at the equator and it grows outward to meet the lateral walls. Outward, see, these are the gradually grown, these vesicles. Due to the vesicles only it make as a one partition. They fuse together to form a cell plate. Here is cell plate. It separates the two daughter cells. Later the cell plate becomes as a middle lamella. This is like a middle lamella. From the vesicle, see here vesicles are formed slowly. They will be continue and separate. Thus as a cell plate. Then it finally becomes a lamella. Due to that, one cell becomes a two cells. That's the cytokinesis of plants. During the cytokinesis, organelles like mitochondria, plastids get distributed equally between these two daughter cells, and if sometimes cytokinesis is not happened, it's absent. Then what happened na? Multinucleated, that's a syncytial stage. It is a liquid endosperm which is the 3N stage we can notice in the plants. Thank you.